lecture is entitled Lifespan and Intergenerational Legacy of Early Childhood Malnutrition on Brain and Behavior, the 48+ year Barbados Nutrition Study. Let me say a few words about Professor Galla before we start. Janina is a professor of psychiatry at Harvard Medical School. She's a senior researcher at the Division of Pediatric Gastroenterology and Nutrition at the Massachusetts General Hospital for Children. She's a summa cum laude graduate of the Sophie Newcomb College of Tulane University and obtained her MD from the Albert Einstein College of Medicine and a residency training in psychiatry and child psychiatry at the Massachusetts General Hospital. Her research involves early malnutrition and long-term effects on mental health and brain development over the lifespan and across generations. She also does multidisciplinary research in animal models of prenatal and intergenerational malnutrition. She has 180 peer-reviewed publications and 2 books on nutrition and behavior. Most importantly for this talk, she is the founding director of the Barbados Nutrition Study. Professor Galla has received numerous awards and grants. She's the first Joseph P. Kennedy Foundation Public Policy Fellow. She is the US Senate Fellow and Chair of the National Institute of Child Health and Development's Advisory Council. She's the Blanche F. Ittleson Award recipient for research in child psychiatry from the American Psychiatric Association. And she received the Irving B. Harris Award from the Society of Behavioral Pediatrics. She's also the Newcomb Centennial Award winner from Tulane University, and she received the Leon Eisenberg Award for Leadership in Mental Health, Developmental Disabilities, and Research Ethics. So once again, we are very, very honored to have a lecture from such an esteemed researcher and clinician. And once again, the title is Lifespan and Intergenerational Legacy of Early Childhood Malnutrition on Brain and Behavior, the 48+ year Barbados Nutrition Study. Professor Galla.
Thank you, Dr. Evans, for this very kind invitation. I'm very excited to be the first recipient of the public presentation, and would like to in particular thank the Mortimer and Vera Gross Fund for support for this presentation. This is a brief overview of what I'm going to be discussing today. I will be talking about the definition and prevalence of childhood malnutrition, critical periods of brain development when malnutrition is particularly important. The major focus, of course, will be the Barbados Nutrition Study, a 48-year longitudinal cohort study, with a particular emphasis on mental health outcomes of early childhood malnutrition. Some new areas that I will also be emphasizing will be epigenetics, which were performed in association with the other aspects of this research, parallel translational studies in animal models, and New Horizons, which includes a very close collaboration with neuroscientists, with the Global Brain Conference and MNI, and the Cuban Neuroscience Center. So first I'd like to talk a little bit about what malnutrition is and how many children globally it affects. Roughly 1 in 3 children in the world experience some form of undernutrition or malnutrition, which is astounding. It's one of the most common public health problems, even in the present age. Stunting is the most common condition, and it really means when children are below median height for age. Wasting, which impacts 45.4 million children, or about 6.7% of children worldwide, includes children who are below weight for expected height for age. Malnutrition is generally viewed as a combination of both stunting and wasting. And finally, although I won't be dealing with it today, 5.7% of children globally, or 38.9 million children, suffer from being overweight, and many of these children come from households where they have been previously exposed to early childhood malnutrition, which has changed the overall metabolic regulation resulting in overweight. The prevalence of malnutrition in children under the age of 5 has declined substantially between 1990 and 2020. And as you can see, roughly 40% of children globally in 1990 suffered from this condition. And in 2020, that dropped to 20%, but it is far below what the expectations were for this century. And unfortunately, with COVID at this point, we believe that these figures are actually substantially higher. I would also like to point out the fact that although I will be focusing my remarks on Barbados in the Caribbean, the values for malnutrition are listed as 8.7% of children, so the number is quite lower than it is in Asia, Africa, and other parts of the world. But again, these figures are probably artificially low and in fact still represent a significant portion of children in this region. This is a recent UNICEF, World Health Organization, and World Bank estimate of the percent of children with malnutrition, children under the age of 5. And again, as you can see, most of the problem centers in Southeastern Asia and in Africa, but again, the Caribbean and Latin America are also represented here. What I would like to point out, however, is that childhood malnutrition is not limited to low resource settings. I would like to point out that in September 6th of 2020, the New York Times reported that roughly 1 in 4 US children or approximately 16 million children were suffering from food insecurity and undernutrition. And although food insecurity per se is not malnutrition, when it is present long enough, it does expose the risk of that child to developing a form of undernutrition or malnutrition. I know that part of the audience today is also Canadian, and I think it comes as no surprise that in 2020, 50.8% of the population in indigenous zones also suffered from food insecurity, which of course is almost 3-fold higher than in any other region of Canada. So again, even within Western populations, there are populations that are at great risk, and children also suffer from the very same conditions that we see worldwide and in low resource settings. Now to move on to the Barbados Nutrition Study. There were a number of reasons that I started to work in Barbados, which goes back to the early 1970s. First, I would like to give a little bit of a background history. In 1966, Barbados became independent. And as part of that independence, the first prime minister recognized that the 2 leading public health issues impacting children on the island at that time were childhood malnutrition and developmental disabilities. There were 2 very well-trained pediatricians on the island, one of whom was Dr. Frank Ramsey, who was assigned the childhood malnutrition issue, and the second, Professor Bertie Graham, was assigned the developmental disabilities issue. And their job was essentially to help the country to overcome these problems. And as part of that, Frank began in 1967 to collect all children on the island who had moderate to severe cases of malnutrition, and to begin documenting these children and following the children. And in 1972, Frank went to Boston, where he received a degree in public health, in maternal and child health. And he went back to Barbados looking at the opportunity for developing some type of ability to monitor children on the island with malnutrition. One of the reasons that there were so many children involved is that until the late 1960s, most children worldwide were not surviving from malnutrition. And in the late 1960s, UNICEF made an extremely important contribution by developing and making available to the world oral rehydration therapy. And suddenly, children who were not surviving the effects of malnutrition began to survive. And we were globally left with a world of survivors without knowing what the long-term consequences of this early exposure was. And in Barbados, in parallel, these children who were surviving malnutrition again were thought to be developmentally disabled, to perhaps have limited lifespans, and very little was known about how to address or look at their subsequent health and development. In parallel with this, I was myself working with animal models of malnutrition and also with human studies in other countries. And Frank and I merged forces and planned out the establishment in 1973 of the Barbados Nutrition Study. There were several factors that were very beneficial to us. Number one, Frank had already identified a group of roughly 2,100 children that had either suffered from malnutrition or had been at risk for malnutrition. Right now, you can see the picture here of our home in Barbados. Dr. Frank Ramsey, who was our initial and founding director, and our current director and co-principal investigator, Dr. Simon Anderson. Another factor in Barbados that was extremely important is the fact that Barbados, even at the time that I started to work there, had a 99% literacy rate. Now, to give you a context, Jamaica, which is genetically very similar and nearby, had a 45% literacy rate at the time that I started my work in Barbados. So there were a lot of advantages in terms of being able to assess and evaluate the children, but also we had a lot of support from the Ministry of Education from the get-go, and this literacy rate was very important and also showed the tremendous support in Barbados by its government of not only child health, but also child education. And finally, another very important feature to Barbados that made it very useful in terms of a place to do this type of longitudinal research is that conditions were relatively homogeneous. The first two slides that I showed were first me in Bridgetown, and a more recent picture that I took actually in 2020 in Bridgetown that show very little variation. A lot of the homes in the most populated portion of Barbados, which by the way is, if you haven't been there, a 15 by 20 mile small island in the Eastern Caribbean. Part of it is actually borders the Atlantic Ocean, so it's not truly in the Caribbean, and the population is approximately 285,000 individuals. And we benefit from the fact that at least early on, the population was relatively homogeneous. And as you can see, the Human Development Index, which defines mortality and morbidity in the population, showed Barbados to be, although at a wealthy nation status, very, very close to being of a middle income status. It is 58 on a list of 189 nations. So what caused malnutrition in the island in 1966 when Barbados became independent? Here are some of the socioeconomic, cultural, and health factors that contributed to these children being malnourished. And these factors still are important globally in terms of causes of early childhood malnutrition. Poverty, first and foremost, an unstable economy in Barbados. At the time, it was a sugar economy. And when sugar markets were high, there was very little malnutrition. And when sugar markets were low, a lot of malnutrition. And that's because we documented that 80% of income in a family actually went to support food and housing. Barbados also has a cultural practice of early weaning to bush peas, which was not advantageous to the developing child. But most important, at the time we started our work, there was no child leave policy, and breastfeeding absolutely was not supported. And so, given the fact that this is a matriarchal society and women are extremely important in terms of bringing an income into the household, since they had to return to work very quickly and there was no support for breastfeeding, children were left to other female members of the household and effectively may have been malnourished. Maternal mental health was also an important factor that we actually reported on very early on. And so women with depressive symptoms who were otherwise stressed because of poor income and lack of support were those very mothers who gave up breastfeeding early and where we saw a lot of malnutrition. And finally, there was a very high rate in the 1960s of infections, particularly diarrhea and measles, impacting very young children. And of course, nowadays, we supplement this with COVID and HIV and other infections that compound problems in this day and age. So these are some of the important factors, and these are summarized actually in a Barbadian calypso song, which is very popular in Barbados, called Endorse the Cuckoo and Give Me a Share, Them Children Ain't Work in Any Way. And what that means is that culturally, because the male comes and goes out of the household, this being a matriarchal society, he is very important, and therefore the food goes to him first. And because the children do not work, they are the last to receive food. And again, another cultural reason for early malnutrition. Now, I would like to point out that in doing, setting the stage for our long-term study, we actually selected from this group of 2,100 individuals, we selected the only children with moderate to severe cases of malnutrition, but we specifically limited cases to very young children who were of normal birth weight, but in fact were no more than 1 year of age. So our study is very unique in that we have an extremely well-documented group of children, and they were malnourished only in the first year of life. This figure, which appears in a review paper that I wrote many years ago, summarizes important concept that that anyone in neuroscience knows very well, which is the fact that the most rapid period of brain growth and development in children occurs between the second trimester of life and year 2, 24 months of age. And it is precisely at this point in time that the effects of malnutrition are irreversible. That's an extremely important concept, and basically it is because the brain is growing so rapidly at this point, and so children are particularly vulnerable at this age. This is a summary of the research design of our study. Again, the study started in 1973, but relied on Frank Ramsey's group of children that were born between 1967 and 1972, and that constitutes Generation 1. And in our Generation 1 cohort, we included 2 groups. There was the malnourished group, which included children with moderate to severe malnutrition, only between ages 0 and 1, and a matched control group. And these were healthy classmates that were matched by age, gender, and handedness. And the reason for handedness is because we knew that we were also going to be including brain measures, and it's important to correct for this factor as well. The selection criteria were as follows. All children were born at the Queen Elizabeth Hospital, which is the largest and only public hospital at this point in Barbados. Their birth weight was normal, so all of these kids weighed more than 2,500 grams, and they had documented prenatal care, they had no birth complications, and they had no encephalopathic events during the course of childhood. So this is basically the setup, and we have information on G0, which are all the mothers of the sample that we studied. And also now we have 206 individuals in the next generation, and they were never malnourished to begin with. One important point that I would like to mention is the fact that all of the malnourished children were included in an intervention program called the NIP, the Nutrition Intervention Program, which was a publicly funded program that followed them from birth to 12 years of age and provided nutrition supplementation, a 2 to 3 day a week nursery school, maternal nutrition education, all the health care that they needed throughout that period of time, and home visits, routine home visits by community health sisters. So these were not simply children who were malnourished and followed. These were children who received what we knew at the time to be the best practices available for children with histories of early malnutrition. Our children with malnutrition basically were children with either marasmus, which meant that they had a deficiency of protein or calories, and that's the picture you see on the right, or kwashiorkor, which are the children with a little swollen bellies, and these are children with primary protein deficiency. And in the Caribbean and Latin America, these two conditions occur very early in life and approximately at the same ages. Kwashiorkor was first identified in the 1930s by Cecily Williams in a pivotal paper in the New England Journal of Medicine. It was recognized in Africa to occur at much later ages, but that is because women there tend to breastfeed much longer. So in Barbados, marasmus and kwashiorkor occurred at the same time, and we included both groups of children. And again, this is just a very quick summary over time of the children that we have followed. There were originally 312 children, and in the next generation, we're down to 206 children. But what is amazing, again, about the work in Barbados is that we know where roughly 95% or more of the population is located. We have information and contact with the majority of the individuals who we have now been tracking for close to 50 years. And this is an overview or a summary of a huge and complex data set that we have collected over the years from infancy now all the way up to 51 years of age in this population, over 7 waves of data collection. We have extensive mental health outcomes on all of these individuals, and I might add that I am trained as a child psychiatrist, and this was my primary interest, but not my only interest. We have extensive data on the neuropsychological outcomes and cognitive functions, medical outcomes, and we have very comprehensively evaluated the environments and socioeconomic conditions in which these children grew up. And more recently, we have expanded to deepen our work with neuroimaging in collaboration with the Cuban Neurosciences Center and Epigenetics in collaboration with Shahram Akbarian at Mount Sinai and formerly at Mass General. So a quick overview of some of our key findings. And again, summarizing 50 years of work is not easy, but I would like to draw your attention to perhaps some of the lead findings from our study. So very early on, when we started to work, as I mentioned before, not only were there very few studies that had looked prospectively at survivors of childhood malnutrition, but we actually did not have what we have now. We did not have gold standard instruments, and we were in a situation of needing to basically create our own. And I was able to do this with the collaboration and assistance of a number of individuals, one of whom was Keith Connors, who has been one of the leading contributors to identifying attention deficits in children and in adults. On the basis of basically using our information from child development schemes and collaboration with these individuals, we created 3 parallel interviews, which were a teacher interview, a parent interview, and a child interview that the child filled out themselves. And we used these interviews as the basis of a lot of behavioral information. And at the first run, when we looked at the teacher interviews and grouped various questions on the interview mathematically into what we call a factor analysis, we were really amazed. This was actually about 1978, to see that what popped up was a grouping of questions regarding attention, memory, distractibility, lack of obedience, restlessness in children that looked very much like what people were talking in Western countries and identifying as attention deficit disorder. But it had never been described in a developing country, and it had never been described in relationship to an early insult, and certainly not to early malnutrition. In 1983, we published the first series of papers ever identifying attention deficits in our cohort of children with early childhood malnutrition. And it is the first report of such a behavioral configuration in a developing country. So the attention, what is important for you to know is that there was a 4-fold increase over childhood in children with histories of malnutrition in the first year of life versus the control group. So roughly 45% of children versus 15% of the control group. Moving on, at age 11 in Barbados, we benefited from the fact that children are given an 11+ examination. This was actually very common in the UK at the time as well, and in all Commonwealth nations. But in Barbados, it was particularly important because there were limited academic seats in high school. And so this was really the great divide. At age 11, you took this exam, and it basically determined what type of education you received thereafter. And so what is important for you to see here is that the children in the comparison in the control group obviously had significantly better scores than the children with either marasmus or kwashiorkor at age 11. And in fact, there were no significant differences between the 2 malnourished groups. And what I would like to point out is that maybe one of the most important papers after the 83 paper that we published was a 1990 paper in which we identified the fact that the teacher reports, the results of the teacher reports at ages 5 to 7 predicted which children at age 11 would not do well on the common entrance exam, on that 11+ exam. So the pathway that we identified at that time was you had a history of early childhood malnutrition. You then developed attentional deficits that impacted school performance. And then at age 11, you were doomed to not do as well on the common entrance on the 11+ examination. And one other comment about the teacher interviews that I failed to make is that, importantly, even though we were seeing attention deficits, the children caught up completely after being involved in the intervention program in terms of their physical growth and size. And in fact, we reported a study many years ago in 1987 in Pediatric Gastroenterology and Nutrition Journal, confirming that the children by adolescence caught up completely, but in terms of their behavior and cognitive outcomes, they did not catch up actually over the lifespan, as you'll see in a few minutes. Again, fast forward, this is, again, an extremely important paper that was published in 2012 in Pediatrics, which showed that those children who did not do, children with early malnutrition, who again did not do as well on the 11+ examination, were also subject to a decline in their socioeconomic status in adulthood. In other words, what happens is that how you do at 11 on the 11 plus exam in Barbados dictates greatly. It is the great divide, and in fact, it limits educational and occupational opportunities over the lifespan. And as you can see, there's an increasing difference between the previously malnourished and controlled children as they approach middle adulthood. And again, this adds even further to the socioeconomic implications, at least across the whole country here, with respect to having had a history of early childhood malnutrition. Recently, in collaboration with CNeuro, we did a follow-up study at ages 45 to 51, and there we documented, again, that 50% of children, of individuals who had had a history of malnutrition early in life versus 15% of controls were subject to accelerated cognitive decline, did not do well on common tests that we now give in most clinical settings and hospitals in order to document whether people are at risk for subsequent dementia. And not surprisingly, the history of early malnutrition and attentional factors were a leading contributor to the poor performance on the MOCA and MMSE outcomes. I think you can also see on this figure that attentional processes are an integral part of the MOCA composite score and also the MMSE composite score. So again, there is an important story to be told. This is again an overview of, I will not go into great detail here, but an overview of the long-term outcomes up to roughly age 50, starting in early childhood. And I think what you can see is there were mental health problems, the most striking of which would have been attention deficits and impaired executive function and accelerated cognitive decline, and an impairment over time of socioeconomic status. In terms of health outcomes, that's still a story to be told. But again, in childhood, there were no differences between the groups. So by the time they reached middle adulthood, there were significantly higher numbers of individuals with metabolic syndrome, and specifically among the women, a much higher rate of diabetes in those who had experienced early childhood malnutrition, which may also be related to the behavioral outcomes. Going on to a quick overview of the next generation, not surprisingly, when we looked at the next generation, and again, this is the offspring of our children who had malnutrition in childhood and the control group, but none of the children in the next generation suffered from malnutrition. And as you can see, these are, this is actually the Connor scale for adults looking at attention deficit disorder, and the two darker columns on your left are the generation 1 results, and the generation 2 results are represented by the two lighter columns. But again, what you can see is that we continue, even though the profile is a little different, you continue to see attentional deficits even carrying on into the next generation. And ADHD symptoms are significant in both groups. So this was, again, an unexpected but extremely important finding. And again, we have many other results as well that presented themselves, but particularly the most striking of which were the attentional deficits even into the next generation. Now, I would like to move on to a related area that may explain some of the reasons for the carryover into the next generation, and this is known as epigenetics. The definition of epigenetics are that these are stable changes in gene expression that arise during development and in response to changes in the environment. But what's important about these changes is that they occur without changing the primary DNA structure. So they are predominantly impacted by changes in environment, and nutrition is one of the major ways in which epigenetic changes occur. This is how we proposed the impact on epigenetics may have explained some of our findings, and I will quickly move into the next slide to show you, again, this is a recent 2016 publication from our group that shows 2 generations of epigenetic changes. And what you can see here is that in generation 1, there are many genes that were impacted by the early history of malnutrition. And however, what surprised us is that there were also changes in the next generation. And a number of genes actually overlapped in the 2 generations, one of which is a gene that's known to be associated with brain development. That's the DPPA5 gene promoter. But what was also very important about this particular publication is that we didn't just demonstrate changes in gene expression in the epigenetics as a result of early malnutrition, but we were also able to demonstrate that 20 of the genes that we observed in the first and second generation were significantly correlated with the ADHD index of these individuals. So what that means is that we were actually able to link the behavioral findings to the epigenetics in these individuals. And that's an important finding that requires further follow-up. And in a more recent series of analyses, we show here the studies specifically limited to 72 of the G2 offspring. Again, these were individuals that were not malnourished themselves, but their parents were malnourished, and they were compared to a control group. And again, we found that, again, the genes that were impacted were significantly associated with attention and executive functions. And also, we found a large number of genes that were impacted were also genes that commonly are related to immune function. Now, I would like to briefly talk a little bit about our translational studies, because they also support the link that we have now reported over the lifespan and across generations with respect to the risk for attention deficits in individuals with early childhood malnutrition. So I've been studying animal models for many, many years, and this is simply an indication of one of our groups of studies, but we have actually tracked the growth and development of rodents over as much as 24 generations following exposure to early childhood malnutrition. And what I would like to point to are really 2 pivotal studies that really beautifully support the work that we are doing in our human Barbados Nutrition Study, in our human studies. And this is the fact that this is a particular study in which we gave animals 2-deoxyglucose, and which documents the metabolic activity of the brain. This is labeled, and you are able to actually track where the activity is in the brain. And what we observed is the fact that there is a reduction in activity in those regions of the brain, predominantly the prefrontal cortex, which is known to be the driver for executive function and for attention in animals and in humans alike. And so we were, again, able to confirm that a history of early malnutrition during development is very specific in terms of where its impact in brain is, and it's directly linked to the kind of behavioral and mental health outcomes that we are describing in human populations. But what was really exciting was a recent piece of work that we have just published, which is along the line of some of the work that is being done at MNI on brain networks. And what we were actually able to document is that the brain networking in rodents that had been exposed to early malnutrition is different than brain networking in animals that are control animals and did not have any history of malnutrition. And specifically, when we tracked brain networks in those rodents while they were involved in an intentional task, the normal animals, the healthy animals, used, again, the prefrontal cortex predominantly, whereas the previously malnourished animals did not use that pathway and in fact relied more heavily on hippocampal formation, which is a region our group has spent quite a long time studying because it is a very good model system of the brain. So again, what is important is that there are opportunities, we think, even with an exposure to early malnutrition, to begin, that the brain naturally tries to find other networks to be able to perform tasks that are required during development. And it's an intriguing possibility in terms of developing and documenting interventions. Now, I'd like to end up with talking about my important collaboration with Pedro Valdez Sosa and his team, and Mitchell Valdez Sosa and the team at CNeuro in defining neural fingerprints of early childhood malnutrition. This group of investigators, as I mentioned before, have been collaborating with our group from the early 1970s. And although the CNeuro Center itself only opened a few years ago, and I was honored to be one of the attendees at the opening of that center, the neuroscience group goes back many, many years. And again, my link to this particular group goes back to Eroy John and a very important history in terms of developing EEG, computer use in the Caribbean, and again, documenting fundamental brain changes associated with early malnutrition. We've been undertaking a lot of important studies, and I'm going to just summarize a few. This is the recent 2018 study, which also included University of Montreal, Dr. Anne Gallagher. And again, we have basically replicated our 1977, 1978 assessments and added to these using more current methodologies. And as I indicated before, one of the important findings from this particular group of studies was the high increase in cognitive decline. This work has also identified that at ages 5 and 11, there were very significant differences in QEEG, obviously in a number of regions that were very specific to the exposure to early malnutrition. And specifically were theta, alpha 1, alpha 2, and beta. And again, I think what is exceedingly important is that the long-term effects of early childhood malnutrition on the brain that we saw both in 1977 at ages 5 to 11, but more importantly, in aged individuals at ages 45 to 51 were mediated by the changes, these changes in brain states that were observed at 5 to 11 years of age. We are currently examining, also with the help of others, including at MNI, that would be Bosch, we are looking at the longitudinal manifestation of changes in brain states over time. And again, what is really exciting and interesting is the fact that everyone, including the controls, had very striking changes in brain states, but the early forms, the early manifestations in 1977 in childhood were highly predictive of many outcomes that we observed, not only in brain state, but also in cognitive state and in behavior at late middle ages. New horizons and next steps. I would like to summarize what the lead contributions of the Barbados Nutrition Study have been. First of all, And most important, our group was responsible for eliminating childhood malnutrition from Barbados by 1980. It's probably the contribution that we are most proud of. And Dr. Frank Ramsey was knighted for this. Our group has confirmed that childhood malnutrition limited to the first year of life impacts mental health outcomes over the lifespan and across generations. Number 3, probably the most pervasive effect of early childhood malnutrition is not on IQ, although that's impacted as well, but the major impact is on attentional characteristics. We observe attention deficits and impaired executive control as a lasting consequence of early childhood malnutrition, even after 12 years of intervention in this population, and even after adjusting for maternal depression, poverty, child abuse, and neglect in this population, because we have very comprehensively also documented these features. Early childhood malnutrition has significant not only health effects, but also has a lot of bearing on economics globally, and leads to a decline in earning potential and social status over the lifespan, and this is actually an increasing decline. Epigenetic changes may be an important underlying mechanism whereby malnutrition exerts its effects. We have also demonstrated in parallel translational studies that the brain regions that are most vulnerable are those involved in attentional processing. And finally, multidisciplinary research, which involves looking at brain neuroimaging and brain biomarkers, are very important in terms of their potential to identify early on high risk and also resilient individuals. And what our important goal is to advance cost-effective and targeted interventions in low resource settings and to use our findings accordingly. Again, this is simply an overview chart showing you the vicious cycle of poverty and how it leads lifelong to malnutrition. It is actually a summary of my life's work, actually, because we have filled in many of the pieces here. Next slide, which will be close to our end slide here. I'd like to emphasize that prevention is a primary goal. WHO talked about this yesterday, but we need to start at the very beginning. But we also need to recognize that nutrition intervention programs alone, while they save lives, do not necessarily improve many of the long-term outcomes that I was talking about. We do need to target high-risk children, but we also need to target attention and executive functions. The earlier we identify individuals, the better. Epigenetic studies are extremely important, but the bottom line is that biomarkers of early malnutrition have to be identified, and that's what we are hoping to do now, and was discussed earlier in our Global Brain Conference. And they must be inexpensive, scalable, and acceptable in low resource settings, because many of the methods that we have are really only useful for those populations where childhood malnutrition is not so prevalent. Finally, it takes much more than a village. It takes so many people to do a study of this type, and I want to extend my thanks to not only everyone on the list, but include many others who probably couldn't fit on this time around. But these are my two primary groups. As you can see, this is our group in Cuba, and on the right you will see our group in Barbados. Without these people and without the participants of the long-term Barbados Nutrition Study, we would not have been able to do the work that we've done. Again, I thank you very much, and appreciate the opportunity to present our work to you.
Thank you very much, Janina. That was a wonderful presentation of a monumental study over nearly 50 years. There are not many studies can last that long and be very, very relevant to today. We have numerous questions to field. I'll take the first one from Ariel Frame. How much does the type of food eaten, lipids versus protein versus carbohydrates, contribute to malnutrition compared to the magnitude or lack of food?
Well, again, the children in our group all had protein deficiency, which is, the building block of the brain, especially during early development. So the type of food is very important for sure. But there has also been a lot of work, certainly in the last 20 to 30 years, on micronutrients and how important they are as well. So again, individual components are important. We opted at the very beginning to address our study specifically to children who either had protein calorie deficiency or abject protein deficiency.
All right. Another question. By the way, anybody who has questions, please put them in the question and answer section on the Zoom link, and I'll get them and read them out. Janina, how much do genetic factors influence the etiology and clinical trajectory of malnutrition? And secondarily, how generalizable are your findings to other global populations experiencing early childhood malnutrition?
So the genetic, again, it is our feeling that the primary influence is really epigenetic, meaning, environmentally induced changes. Obviously, there may be genetic factors as well, but it was not the prominent finding in our population that. Again, this is a global problem, and undoubtedly there's probably some families that are more vulnerable than others, but the primary reason is really more the environment in which the child and the family is placed rather than genetic factors per se. In terms of replicability and relationship to other parts of the world, we are actually at this point in time hoping to move this study forward to be able to replicate elsewhere. But there is now very good evidence from other groups as well that the attentional findings that we reported so many years ago, in fact, stand up in many other settings as well. And so we are able to observe this. And but again, there are really no studies that have looked as comprehensively as we have. And since we have incorporated behavior health, neuroimaging, and epigenetic outcomes, in all of the individuals in this study, so each of the various studies that have been done in different settings has contributed other components, but in general, it does appear that the main findings from our study hold up in other settings as well.
All right, the questions are starting to pour in, Janina. How can we be sure that the effects we observed are attributable to child malnutrition, given the myriad of challenges these children face? Poverty, crowding, infections, maltreatment, etc. Couldn't there be a number of other causes?
So again, this is an interesting and important question. We, from the very beginning, realized how important it was to document the ecology and the environment, details about the environment in which the children were growing up, which included but was not limited to maternal and parental mental health, which had really never been looked at in these types of settings before. All of our work looks both at unadjusted models, which would mean individuals that have been subjected to exposures across all of those risk categories. But we also have adjusted models in which we are able to step by step control for and adjust for each of those particular variables. So for instance, we have very carefully documented maternal mental health and We adjust for that. We have very carefully documented for early maltreatment histories in these individuals, and have a series actually recently of about 4 publications looking at the independent and conjoint effects together with malnutrition, separate from malnutrition, et cetera. So it is necessary in a study of this type to document, but our studies have demonstrated that even when you control for socioeconomic factors and other adversities in the environment, the finding of impaired executive function and attention deficit holds up as a very striking outcome.
We have 2 questions on this, more or less the same topic, from Ina Puce and Lee McIntyre. Are there any differences in brain development between male and female children as a function of early malnutrition?
So again, that's an important, again, a very important question. I will tell you that early on, I was involved in a study. My mentor actually started a longitudinal study in Jamaica, which was limited to males. And the reason for that is because beginning in 1950s with the papers of Passamanik and Noblick, et cetera, there was some notion that male fetuses were more vulnerable to the effects of prenatal, but also postnatal malnutrition, and that there was no point in really looking at the female developing brain. My mentor actually, when I was a first-year resident, passed away, and I was in a situation of needing to pick up and continue this work, which I clearly ended up doing. I really felt that it was unconscionable to not have the ability to be able to compare brain development across the genders. And consequently, we, for that and many other reasons, abandoned the work in Jamaica and started up afresh in Barbados, including both males and females. What is interesting is that in general, the females in the Barbados population did substantially better than the males did on cognitive tests, and seemed to be more resilient to some of the effects of early childhood malnutrition. But again, after you control for the myriad of early exposures, there really are very few, if any, differences in the outcomes between the 2 groups. And we are not observing major gender differences at this point. But again, it's a work in progress, and this is part of the work that I'm doing at this point with C-Neuro as well.
Okay, 2 questions from Faranak Farzan. Very impressive work. Is it identified how many generations following the first generation of malnutrition would be impacted?
So I've always been intrigued by this because, you know, if anyone, not that this is a major persuasion of mine either, but, you know, the Bible says 7 generations, right, of, you know, things lasting. But one of my early studies was, I actually inherited from Reg Stewart at the London School of Tropical Hygiene and Medicine, a colony of animals that at the time had 13 generations of malnutrition. I brought the colony over actually on July 4th, flying on what was then a Pan Am jet, first class transport from my rat colony. And I brought them, they lived at MIT for a number of years, and we kept them going for 25 generations. And what we documented is that brain and behavioral effects of early developmental malnutrition in the male rodents lasted for up to 4 generations. After the 4th generation, they normalized. For females, it was 2 generations. So again, some evidence of less of an impact, you know, in the females reared under identical conditions, and 4 generations in the rodent population. We don't have, obviously, that ability to be able to document in the human population over that period of time. But again, we're now actually on to the 4th generation with our Barbados study, so we may have some answers.
It's a fascinating observation. And also, are there currently interventions that can be applied to reduce the impact of G1 malnutrition in subsequent generations?
Yeah, so again, there's a lot of very important work looking at various types of intervention, including early childhood stimulation, which was the subject of the Grantham McGregor work in Jamaica, which is an important study that showed that some of the behavioral and health impacts of early childhood malnutrition were reversed by having access to once-a-week stimulation at the age of 2 to 3. Now, this was a somewhat different group. These were children who were not as severely malnourished, and the ends were very small. But it is noteworthy that the attentional deficits, even with this intervention, were problematic. So I think right now best practices, would suggest that early intervention, some form of stimulation, as well as nutritional support, continue and seem to be optimal. But I think also it's important to recognize that interventions that are focused more specifically at executive functions and attention seem to be the most useful and the most helpful. And some of those early interventions were not specifically focused. So I think that's where the money is. I think we need to go where the money is, and I think we need to focus on supporting prefrontal cortex functioning in these populations. I think that's where we're going.
Okay, just a couple more. And Louis Doré-Sava has asked, the ADD problem has been identified for a few decades. Is there now a specific intervention in place in the Barbados cohort to address this, despite the low economic status of the population?
So what's really interesting to me is that, again, it is not exactly the ADHD problem that we see in the Western world, in that, at least in the G1 population, the primary clinical characteristic was inattention in the population. And we've often been asked, well, did you medicate these children? And the answer is no, we did not. And we felt that that was unethical. And I still feel that that's unethical. But we were able to, fortunately, in those days, work very closely with the Ministry of Education in encouraging smaller classrooms, particularly for children at risk in the earlier years. And that seemed to be very helpful. Now, again, I think there have been, political and socioeconomic changes in the island, and the continuity of these changes may not be there, but that's been one very important intervention that we feel actually has been helpful, and an important contributor. But again, our primary intervention, even in that setting, has been to change policy and to change educational practices, and this seems to have been a very useful strategy.
And lastly, not a question, a comment from Giuseppe Chiarenza. It's not really a question. These studies support greatly the epigenetic probabilistic model of development of Gilbert Gottlieb. It's, I think, a nice point to close on.
Alan, can I add one comment to this? Okay, so it absolutely does, and Gilbert Gottlieb many years ago actually taught me to ski at one of the early winter conferences, and but absolutely, a lot of this work is would not have occurred without his major contribution to our understanding of brain development.
Okay, I think that ends the question period. Somebody asked, will the presentation be available? Yes, it will be. It's all been recorded. And I would now like to thank Janina for a wonderful presentation. I think you've really stimulated the audience with a lot of questions and a lot of interest. And I think it's been an exciting presentation. I should say that this is also a very good link to the activities of the Global Brain Consortium, which preceded this lecture. Janina is leading one of the work projects within the context of the Global Brain Consortium to look exactly at this subject of malnutrition and its impact on brain development. So please do, if you have the chance, look up on the Global Brain Consortium and find out a little bit more about not just this project, but the wider activities of the Global Brain Consortium. And with that said, I'd like to thank Janina once more. And I'd like to thank everybody for organizing this conference. I think it's been very entertaining for all of us. And thank you very much. Bye-bye.
Thank you.